All right, guys, we're back. We're going to do one of our lectures now as part of our immunology on antibodies. With the different things we've been talking about through the program, I know you've heard about antibodies, and I drew you some pictures. Oh, my pencil's not wanting to work. There we go. Our antibody looks like that. That's how we designate it right now. And so let's move along with this. So why are antibodies important? And I find it funny. We say antibodies, antibodies, how different people pronounce it. Anyway, they are a key in helping us fight infection. We also know how big they are right now. What a big deal they are with this Mr. COVID, okay, causing problems because we use the antibodies to fight it off. We use the antibodies to share to another individual to help fight it off. And we're trying to develop that vaccine, okay, to inject into people so they make the antibodies. Okay? So that's one of their functions, fighting infection. It's important. Well, what else are they important for? Diagnostic tests. Okay, we are using these antibodies, specific ones, for a lot of our testing. We'd be lost with our immunological tests if we didn't have these antibodies. We use them to diagnose the infection. So we look to see if the patient has the antibody. We're using the specific antibody in the test method to look for the antigen in the patient. So we're looking to see if the patient here, okay, we're going to draw HIV in him, that little triangle. And so we're using the antibody to HIV in the test, okay, to see if he has the antigen, the virus, okay. What else are we using it for? It's used to find an antigen in a patient, which is what I just talked about there, okay. Disease testing, cancer diagnosis. So we know certain cancers have markers on them, and that's what we're looking for. We use antibodies for pregnancy testing, and I have an image of that to talk about again, which, we visited that in our intro course, but now that we've learned multiple things, we're going to go back and review it, and hopefully the puzzle pieces really fit together a lot better now, okay? Drug testing. And basically what we're using these in drug testing is when you, let's see, this guy here smokes pot, put a little thing in there, smoke goes off there, okay, goes into the body breaks it down into little analytes as well to try to detoxify your body from it and excretes it in the urine. Okay? And so we use a test method with a specific antibody to those metabolites, is what they're called. Okay, MET. Okay, so that's what we're looking for. Okay, they're utilized in research in biotechnology. Okay, for sure, because of drugs. We're actually making drugs, and that's where those HAMA antibodies come from as well. These are the human anti-mouse antibodies that cause problems in some of our test methods. So here's that picture I promised you with the pregnancy test. And so right here, this is our sample, and this really should be yellow, right? Either way, whether it's urine or serum or plasma, it should be yellow drop going on there. Okay? And so what we're looking for is the beta HCG, okay, unit to determine if someone's pregnant. And so what we have here are specific antibodies that are in here, okay, to that HCG, and they are yellow. Okay, the green here is the HCG. That's the HCG. So if it's present, this antibody binds it, and then this antibody here is specific to it as well. So they both bind here on this line. This is the test line, if you remember. Okay, so it's a sandwich. And when these two bind together in this complex here, you see that line. Okay, this one here is stationary, and while this one is mobile, it's called a mobile phase. And so as it migrates across there, if there's any of this left, which there should be an excess of it. Okay, it's specific for this marker here. Okay, and so it's going to bind on there, and we're going to have, this is our control line. And remember, when you have your pregnancy tests, there's your test line, and it usually is marked with a T, and your control line. And if you don't have a control line, it's invalid. Okay. And basically, that's what this is designed to do. It's designed to make sure that everything that you put on there has migrated the whole way across the test so that it worked accurately. And here's a rapid strep. And it works the same way. We're just looking for a different antigen. In this one, we're not looking for beta HCG. We're looking for an antigen from that streptococcus organism. Okay? And remember, what does streptococcus look like? It would be in a strip. Okay? It'd also be gram-positive cocci. Okay? Giving you some, making you think and remember again. I know you hate me for that. It's okay. So here's your test line. And again, here's the antigen from the swab. Okay. So this, we put the test strip in there. As it migrates up here. Okay, here's our mobile antibody. And see, it's got this little tag for that color. Okay, it attaches to the antigen. And this antigen attaches, this is the stationary antibody. And that's where you get this line. If it's not present, okay, this continues. These are just some other things that are in there to make sure it's, it says red tag and blue tag down here, how these guys work. So we want to make sure that the fluid has migrated clear to the top for the test. And so when this one attaches to this antibody up here, this is your control line. Test line, control line. Okay, little different theory behind it because you have the red and the blue tagged ones. Okay, then the pregnancy, but really the same. Okay, so what is serology? It's the study of that reaction, okay, and the properties of the serum components of the blood. It's not chemistry because we're not measuring those analytes, but what we're dealing with now primarily is antibody and antigen reactions. And what's in vitro, of course, I always just think of, in vitro fertilization, and that's in the test tube. Okay, so there's our test tube. So we use it to detect antibodies. Here's that Hep B. We use it to look for that antigen, HIV, and etc. There's even more. Okay, Lyme's disease, and that's just a few. Right? Oh, let's take a look at this. This should bring back a little bit of memory. Okay, this was our SPE, serum protein electrophoresis. Okay, and remember this gamma region. Okay, so we're coming full circle with some things. We're really trying to tie them together. This gamma region is where our antibodies are, and we call them, again, here's not N. Okay, gamma globulins. Gamma globulins, immunoglobulins. Spelled this one wrong, we'll fix that, okay? Immunoglobulins and antibodies. And you'll see that abbreviated with the AB, okay? But they're in this region here. And we'll also relate to this region when we talk about multiple, I keep getting ahead of myself on this one. I keep spelling it wrong everywhere I go today. Multiple myeloma. Okay, and I have a video posted for you guys to watch about that. Here's a fun little picture I found. This gives you a depiction here of an antibody. So here's the antibody, okay? And we're going to talk about the different regions of this antibody. But right here, this is the region that is specific, okay, to an antigen, and this is showing you here that area where it's specifically binding to this antigenic site. Okay? And it's just a picture of this organism. These would be kind of fun to play with if they were magnets. It's kind of what they look like. Okay, so here we have an image of our antibody, and we're going to talk about these regions. Okay, and they can be broken down. These have different components, and they are held together by disulfide bonds. And the reason why that's important to be familiar with is because there are different methods we'll talk about that will break these bonds, and then we'll get different pieces. Okay, so this right here, use a different color. We're going to look at the heavy chain. Okay. Goes down through here. Okay. Here's our light chain. Okay. What I want you to see as well is here's our variable region. And our variable region is out here. Okay. And here's our constant region. So this is the same. Okay. So if this is an IgG for, let's just stick with our chickenpox. Okay. The part that's going to be different is here. Okay. So between our chickenpox and our coronavirus, this area right there will be what's different. When we talk about the constant region there, that's going to be the same. Okay. So that's where constant, variable. Okay. And so here's a picture of our heavy chains and light chains. Okay. And one thing I'm going to touch on here is it's in our other PowerPoint as well when we start off. These light chains can be kappa or they can be lambda. Okay. Kappa and lambda. And we don't really work a whole lot with distinguishing between those, but just be aware they are slightly different. Okay. And this one shows that this is after treatment with urea. It's been broken down into those chains. Okay. Heavy and light. Oh, I did move it here. Good. There's our kappa and lambda, so our light chains. And you see it has no functional difference. Right? And they are roughly present in a 2-to-1 ratio. Those are just some facts that, occasionally they like to throw on tests to see how much you know about them. Okay. These can be these light chains. So what happens with these? Remember, I wanted to get back to multiple myeloma. All right. This is where the body is the patient, they are producing what we call plasma cells. Too many. Okay, this is like a cancer. And so the patient is producing just gobs and gobs and gobs of plasma cells. And this will look very similar to a picture you'll see in the video. Right? That's what they look like. And these plasma cells, if you remember, when a B cell becomes activated, it turns into a plasma cell. And these B cells, remember, they have a memory. Okay, and B cells, when they turn to plasma cells, they secrete antibodies. Okay, so now in multiple myeloma, these plasma cells here have just gone crazy. Okay, and what they do is they secrete tons and tons of antibody and light chains. And you're going to see that, okay, in this video too. And I believe they even say it's usually specific to IgG or even IgA, okay, depending which one it is. They're going to be exactly the same because remember, cancer cells clone themselves. So, but they also have those light chains. And so we find this excess, and remember, these are made up of protein, okay? We're going to find this in the urine. Okay? One of the first diagnostic markers of this tumor are Bence-Jones proteins, okay? And we find those in their urine. And if we do serum protein electrophoresis, boy, I'm going to do really bad on this, because this is supposed to be big here, we'll go across here, and this is supposed to go along like that. Multiple myeloma, remember these are antibodies, okay, and we're producing an excess of them. So in serum protein electrophoresis, this is where we get that monoclonal spike right here. And I don't remember and haven't found in any of my reading, they call it the M spike. Okay? And so this is a cancer. And because we're producing too many of these that really they're not really helpful, they're not specific, it crowds out all of the other cells in our bone marrow, so we're not producing the antibodies that we really need. Okay? And here is just, you might see this term, okay, as opposed to the constant region and the variable region. So your FAB, this is fragment antigen binding, okay, because this is the part that binds the antigen, okay, but that's also the variable or antigen binding. And then here where you see FC, we call that the fragment crystallizable. Okay, or constant. Okay. So you might see those different terms. And again, here's just another picture of your heavy and light chains. And you can see this heavy chain is pointing out they both have this variable region on them. Okay, and then the light chains are either kappa or lambda. And this is showing you that it's coming from the B cell. And it's this B cell, because of this little halo right here, really is acting as that plasma cell. Okay. There's your plasma cell. Affinity versus avidity. What I want you to do, open this up and watch this video. It does help. Affinity is the overall fit. And let's see if I have that. Yeah, the picture right here. So the affinity is the fit. So you can see how this guy is like a perfect fit. It's single, okay, combining site. And over here, it fits in there, but it's not a perfect fit. So this one is definitely a higher affinity, and this one's lower. Okay. Now, avidity is the overall strength of the binding between antibodies and antigens. So this is single, and this is multiple. And I think I should have added, yes, here's my better picture of that. Okay, affinity, you see in a nice little fit. Okay, and then avidity with this one, this antibody here has a much higher avidity because it's actually bound, okay, for those sites, where this one just had two. Okay, I know, why do we have two different words for that? We'll use it a little bit more in blood bank. Okay. There are 5 main classes of antibodies, and you can see here they are. I have them listed, and we will go over these. They're separated. Oh, that's funny how that box went. Let's just see if we can get a nice little circle around there. No. Okay, structurally they are similar. So they all basically start out like that, and we're going to see how each one's a little different. Okay, so here's an IgG. See that? This IgE is a little bit longer. It has 1, 2, 3 of these. Here's our D, pretty similar to our G. We've got some binding going on right here that's a little different. One of the big things to remember is this IgM is a pentamer. Okay, There's 5 of them. 1, 2, 3, 4, 5, which also means because each one of these have 2 binding sites, there are 10 binding sites. Okay? These guys all just have 2. This one has 10 binding sites. And here's an IgA, and they pretty much show this, usually together with 2. And they have this protein that wraps around to keep it together. That's IgA. And we'll talk about some different characteristics of each one of these. So IgG, this is the one we have the most of. Okay? It is 80% of the immunoglobulins that you would find in serum. Okay? It travels to extravascular space. That's important. Okay? Helps fight infection. Opsonizes. Remember, it prepares it to be eaten. Activates complement. This is important because what does complement do? Okay, it causes cell lysis. So when we have this cell, and it's a bad cell, and that antibody gets on there, it's going to activate complement, which is a whole cascade, which will lead this to that literal rupture of that cell, lysis of it. Okay, we'll see more of that complement. We have a whole nother lecture that goes along with this. Neutralizes toxins and viruses. So it blocks the binding. Okay, so here's this little toxin, okay, and our antibodies find it, okay, kind of cover it all up, and now this toxin, it can't get into whatever cell it's trying to get into. If it's a virus, okay, the antibody coats it, inactivates it, and now that virus can't get into the cell it wanted to infect. Okay, so they work by that. Enhances clearance. So now that this, I didn't know we put an eraser on. Now that this guy's covered with antibodies, as it flows through, remember our spleen, all right, as it flows through the spleen, it's going to clear it out of our circulation, get rid of it. And here's some more pictures. Yes, they're a little bit better than mine. Okay, agglutination. Okay, so it can bind to 2 of these. Okay, it's going to make 3 of these clump together easier to either phagocytize or remove through the spleen. It's going to make it easier to get rid of 3 at a time, less work than 1 at a time. All right, here's our opsonization, and we can see these antibodies have coated it. Our macrophage or dendritic cell recognizes that, eats it up. Okay, down here you can see, I thought this was a good picture. Okay, neutralized it so it can't infect that cell. Okay, and here's the toxin. It blocks that binding. Okay. Here's a eosinophil, antibody-dependent cytotoxicity. So this eosinophil has these little antibodies that are attached to it. So these antibodies are on here, and it recognizes, here's an epitope. An epitope is basically the antigen. Okay. So the eosinophil recognizes it, attaches, and then spits out these cytokines, porphyrins, enzymes, whatever it has in its arsenal to destroy that larger cell. In this instance, it's showing it as a parasite, because remember, eosinophils are against parasites. And up here, here's the complement activation. And this is their depiction of the whole complement cascade. They're putting it in this one little thing, and it's causing the lysis of that cell. Kind of fun. IgG does have a long half-life. This is one thing that's particularly very important, and you're going to get a lot of in blood bank. IgG crosses the placenta. Okay? So for good things and bad things. So it crosses the placenta, sharing immunity. The problem is when baby is different than mom, and mom actually forms that antibody that's against the baby. And so when that goes across the placenta, and it is directed against the baby. That's where we get hemolytic disease of the fetus and newborn. Okay? And here's this fun question. So, IgG is mom's, okay? And we haven't talked about IgM yet. Okay, we'll talk about that in a little bit. But if we find IgG in a baby to rubella, it's from the mom. Okay? Like that, it's mom's. And IgM to rubella, this is going to be the baby's. Okay, and we'll understand that more when we talk about the IgM shortly. Oops, sorry about that. So IgA, it's the second highest in serum concentration. There's little function in the serum where it doesn't do much when it's in there. Its primary function is in secretions, okay? And this is where it changes to that dimer, okay, which you saw had those two hooked together with that protein on there, okay? And this, again, they're in secretions. So this is tears, sweat. Also, it's very high concentrations in breast milk. Okay. It works to help prevent the entrance of pathogens. What else do I want you to see from here? Easily removed by ciliated cells. So it attaches to that and then attaches to these mucous membranes, okay? And then we get rid of them. Sneeze, cough, so forth, okay? Blocks adhesions of bacteria. So if you think about that, remember some of that bacteria, and it has those little pili on it, okay? And that's supposed to help it. That's one of its virulence factors for adhesion. So if we have these IgAs, it's going to work to neutralize that so that bacteria can't attach. Okay? Does not bind complement. Does help opsonization. Okay? Not transferred from the placenta to the fetus, but it is in breast milk. Okay? And there's that's a much better picture than mine. Okay? Now, IgM. This is that pentamer that I told you about. It has 5, 2 heavy chains and 2 light chains. So that means it has 10 binding sites. Okay. It is 5 to 10%. Starfish-like pentamer. You see it both. This is actually when you are exposed to, let's just stick with coronavirus. IgM is the first antibody produced. Okay? And then that leads to IgG, which is more specific. Okay? And your IgG is part of your memory. Okay, so when you are exposed again to coronavirus, you're going to produce IgG as well. Okay, so IgM is your first, which is why when we were measuring that in the baby, okay, we'll put a little clearly cute hair on it this time. When we were measuring it in the baby, the IgM we were able to say was the baby's, and the IgG that was present was the mom's, because the baby's just born, so it was just exposed. And IgG is long term. Here's a nice picture, and here you can see all of those fabulous binding sites. Okay. We love IgM and blood bank. Multiple reasons. This is the largest, and you can see why. It's 5 of them. It is called the macroglobulin because it's so big. Okay. Here's our 10 we talked about. Okay. Can be flexible. So if you think about that, it can twist a little bit. Yeah. It's got some good dance moves, right? It is the best one at agglutination and precipitation. So where you'll see this is we're going to use it for some of our agglutination tests. And if we think about it, if we have this guy that comes out, and we'll see, like I said, way more about this in blood bank, and we attach this, and he has one attached here, and here, and here, and we have another one of these guys here. You can see we're creating this huge network now, and that's really what it is. Okay, we're going to attach one here. We'll have another network here, more, where if this was just IgG, it's going to take a few more to create. Okay, you can already see this clump is bigger than, say, this one. Okay, and let's add one more antibody just to keep these equal, so you can see now that we even have, this one would be 1, 2, 3, 4, oops, I have 5 there, this 1, 2, 3, 4, and I'll add one more just to make it complete, and there's 5. Okay, and so this has the same amount now, but you can see where that network's much larger, so we can visualize it. Okay, this guy, we're gonna remember this. He is very good at fixing complement. So if he fixes complement, he's going to cause the lysis of those cells. Okay? So here you can see again, he is our first immunoglobulin produced. Okay. First one produced in a newborn. Of course, if it's the first one produced, it's the same there too. Okay? It does not cross the placenta. Okay, it is so big. Okay, it doesn't cross the placenta. Okay, that's important to remember. It is on the surface of B cells. Let's see, what did I want to make sure we remembered on here? IgD is too, but so these two are on your B cells. Okay, so this is where you have your B cell. Whoops, I don't want a line there. Your B cell. And we did see some pictures of this. So what happens is this guy comes out. All right, and he's just looking for something bad. Okay, and then when he defines it, that's going to bind to it. It's going to trigger your signals. It's either going to take it and have it opsonized. Okay, it's going to phagocytize it on its own, or it's going to take it to a dendritic cell. Different things. Works different ways. Right? IgM, you can see where it sends those signals to B cells, and this is important because when it finds something, if that IgM is out there and it finds this guy, it's got to send a signal out, because remember, your B cells, it's not attached to all of them. We've got some free floating too. And this B cell comes and says, oh, yeah, I remember this thing. And I have more antibodies I can make to fight it and neutralize and take care of or remove the other ones that are floating around in the circulation. Okay? And here's another picture how IgM can attach. See, these are specific, shows different ones. So this would actually be a different IgM antibody than this one because they attached to different sites. Okay. IgD, we have a very small amount of this. It's on the surface of mature B cells. It does function as a specific antigen receptor. We don't know tons about IgD, and we're learning more. But it doesn't bind complement. It does not. It's not opsonic. It's not an opsoninogen, and it's not transferred to the fetus through the placenta. So we still got a lot to learn. Okay, IgE. We're still learning things about it, but what we know for sure is it has a huge response here in hypersensitivity reactions. What hypersensitivity reactions lead to is anaphylactic shock. Which is bad news, okay? Which can lead to death. Okay? So IgE, it is in the lowest concentration, maybe of the lowest concentration, but it has some dramatic effects. Okay? So it's made by plasma cells along the respiratory tract. If you think about that, it's allergies. It's where we breathe a lot in. Okay, digestive tract as well. Binds to mast cells, and if you remember those mast cells, they spit out a lot of histamine and heparin. Okay. Oh, there. Yep, I have it listed right here. Histamine, heparin, and chemotactic factors. Those chemotactic says, hey, I need help over here. Send more over. Okay. Classic allergy symptoms: hay fever, asthma, vomiting, diarrhea, hives, shock, and anaphylactic death. Okay, so it can really go full circle, full force in the wrong direction. Okay, so first exposure, here's we're going to show the allergen here, and this is why most of the times when you're initially exposed to an allergen, you don't have an anaphylactic. Okay? Like a lot of people will say, I'm not allergic to poison ivy. Well, the first few times you touch it, you're probably not. But each time you touch it, the reaction actually gets worse. Okay, so here's the first exposure. It enters the body. Okay, second, step 2, produces some large amounts of class IgE against the allergen. Right? IgEs attach to the mast cells when found in the body. Okay, and here's where it starts spitting out these granules. And then your second and third, because these guys have a memory, okay, with each subsequent exposure, it gets worse and worse. That's the concerning thing. And here it's showing you, here's the allergens. These guys are ready, okay, because it knows, it recognizes it. And the reaction is much more significant. Okay. Leaky blood vessels. Oh, that sounds awful. Okay, fluids, and there's the tissue swelling. And you could see, or if any of you were allergic to bees, okay, the more you're exposed, you usually get that eye that's huge swollen. You might see someone with those huge lips, okay. They talk about, I know, it's not a very good picture, their throat starts to constrict because of the swelling, okay, and they have trouble breathing, and that's what happens with your anaphylactic shock. Okay, thanks guys, good job.